Dash group of animals are diploblastic. Coelenterata and Ctenophora. And which group of animals are triploblastic? From Platyhelminthes to Chordata. And notochord is derived from which germ layer? Mesoderm. Next, Evelyn.
Yes, sir.
In which group of animals neurons first appeared?
Neurons first appeared in Nidaria.
Cnidaria or
Coelenterata. Yeah.
And first diploblastic animals.
Nidaria or Ctenophora.
Cnidaria and Ctenophora.
Ctenophora.
And in which group of animals we can find cephalization?
Platyhelminthes.
Dash are acoelomates.
Acoelomates. Sir, should I give the examples, sir?
Acoelomates, Platyhelminthes.
Okay.
And even we consider Porifera and Cnidaria as acoelomates. But the triploblastic animals who are acoelomates, example Platyhelminthes. Dash are pseudocoelomates.
Ascalmenthes.
And the first eucoelomates.
Annelida.
And which group of animals have mesoderm scattered in the form of pouches?
Ascalmenthes.
Dash group of animals have body cavity lined by mesoderm.
Annelida.
Right. Next up. Where are the other person? Ranika, I think you are not attending yesterday's class, I think, isn't it?
I attended yesterday's class, sir.
But I did not see you yesterday.
Because I did not on my videos.
Then you just give the answers. What is the level of organization found in Porifera?
Cellular levels.
Cellular level. And what might be the reason for loose aggregation of cells?
Due to the lack of nerve cells.
Hmm?
Due to the lack of neurocells.
Due to the lack of nerve cells. And what is the material found between ectoderm and endoderm in diploblastic animals?
Mesoglia.
And in open type of circulation, the blood generally passes through dash.
Capillaries.
Open type. Passes through the body cavity or sinuses. In which group of animals we can cut the animal into two equal halves from any plane passing through the central axis?
Radial symmetries.
And we can cut the animal into two equal halves only through one plane in?
Bilateral symmetry.
Very good. Then Hrithwik, Ritwik, can you hear my voice?
Yeah, sir. Yeah, sir.
Then you turn on your video.
So because of the internet issue, I could not turn on the video.
And who is Derwin? Derwin. And Levin. Are you there in class?
Yes, sir.
Yes, sir.
Then turn on your video. Right, Ritwik. I mean, Derwin. Who is Derwin? Right. Next, let us see. Right. Now let us start different phyla. Now remember, in every phylum, we have non-chordates. That is not writing properly. We have non-chordates and chordates. Non-chordates and chordates. What is the difference between non-chordata and chordata? In non-chordata, notochord is absent. Notochord absent. Right. In non-chordata, notochord present. In chordata, non-chordata notochord absent. In chordata, notochord present. I told you notochord is derived from which germ layer?
Mesoderm.
Mesoderm. Okay, ma? Next. And we have phylums from Porifera to Hemichordata. We considered all these as non-chordates. Some consider Porifera to Echinodermata as non-chordates, and Hemichordata is a connecting link between chordates and non-chordates. And we have phyla like Porifera to Echinodermata in non-chordata, and in chordata, notochord is present, in which we can observe different classes. And what are the different classes and different groups which are found in chordata? We'll see. I think you might be aware of the familiar classes like fishes, amphibia, reptilia, aves, and mammalia. Now, while reading this particular non-chordate group, will frame certain rules or will follow certain sequences in order to get that particular subject easily. And what is the sequence generally will follow? I'll tell you. Remember, whatever the phyla you take, whether it is Porifera, Cnidaria, Ctenophora, Platy, Aschelminthes, Annelida, Arthropoda, Mollusca, or Echinodermata, what it may be, will frame certain points. And we just will follow those points in each and every phylum. What are those simple points? I'll tell you. If you are aware of these points, this sequence, you can remember any phylum. I'll tell you thing. Number one, introduction. Number two, habitat. Habit. Levels of organization. Symmetry. Coelom. External features. Internal features. And then physiology. In physiology, generally we'll discuss the points like locomotion, digestion, respiration, circulation, excretion, nervous system, sense organs, reproduction, and development. This is the sequence we have to follow while reading the file. I'll repeat once again. Introduction. Introduction. Habitat. What is habitat? The place where the organism lives is habitat. If you remember these points, you can recollect any phyla among the invertebrates. The place where the organism lives is habitat. How they live, habitat. Levels of organization, cellular, tissue, organ system. Symmetry, coelom. External, internal features. Then locomotion, digestion, respiration, circulation, excretion, nervous system, sense organs, reproduction, development. This is the sequence we have to follow. I'll explain each and every phylum according to this sequence. Let us start with Porifera. See how simple the phylum is. Number one. Amma, what is the first pointer? Introduction. Poriferans are commonly called as sponges. This is introduction. Then habit. Mostly marine, few freshwater. Few freshwater. Then next, levels of organization. Cellular level. Cellular level. Then symmetry. Asymmetry. They don't have any body cavity or coelom, so they are acoelomates. Commonly called as sponges, mostly marine, few freshwater. Example for this freshwater form, spongilla is a freshwater form. Mostly marine, few freshwater, cellular level, asymmetry, acoelomates. When you look at the sponge, this is sponge, where the cells are arranged in two layers. Outer layer is called pinacoderm, inner layer choanoderm. But they are not diploblastic, remember. Inner layer is having flagellated cells. Inner layer having flagellated cells. The name of the cells choanocytes, so the name given as choanoderm. They're having collar-like shape, collar-like structure. So the name given as collar cells. And very important, they have small openings all over the body. They have small openings all over the body. These openings are called ostia. They are inlet openings. All over the body, ostia are present. And very, very important, they open into canals. This is canal. Very, very important. The peculiar system found in porifera is canal system. And these canals open into a space called spongiocele. They open into space called spongiocele. And this spongiocele open out through a big opening called osculum. Very, very important. The pathway of passing of water through canal system, ostia, canals, spongiocele, osculum outside. Now, what is the function of a canal system? And this pathway of water flow helping bringing the nutrients along with the incurrent water. So it helps in drawing the food material through water. Number two, along with the incoming water, oxygenated water can come in, help in respiration. Number three, undigested waste materials can be sent out through osculum. Very important. What are the functions of canal system? Number one, nutrition. Number two, respiratory gas exchange. Number three, removal of waste material. At the same time, the body wall also consists of skeleton. Body wall also consists of skeleton made up of skeleton may consist of calcium carbonate spicules. What are spicules? Small spines. Or spongin fibers. So the skeleton may consist of spicules, small spines made up of calcium carbonate, or in some it is made up of fibers made up of a material called spongin.
Excuse me, sir.
Yes.
Sir, can you repeat this water canal system
Yes,
again,
yes.
sir?
I'll repeat once again. I'll revise the points once again. First one, introduction, commonly called as sponges. Number two, habitat, mostly marine, few freshwater. Habit, all are sessile. We just change that point, sessile. Habit, all are sessile. What is meant by sessile, Amma? Stationary. I'll repeat once again. Commonly called as sponges. Number two, mostly marine, few freshwater. Habit, all are sessile. They cannot exhibit movement, but larvae can exhibit movement. Coming to levels of organization, cellular level, asymmetrical, acoelomate. And all over the body, we can find small openings called ostia. The inlet openings are called ostia. They lead into small canals. These canals finally open into a space at the center called spongiocele. That open out through osculum. Now, the unique system found in porifera is water transport or canal system. Water transport or canal system. Now the canal system constitute ostia, opens into small canals, open into spongiocele, and finally open out through osculum. So what is the direction of the passing of water or the pathway of water? Ostia, canals, spongiocele, outside through osculum. Now, what is the advantage of canal system? Number one, collection of food. Number two, respiratory gas exchange. Number three, elimination of waste materials. And what are the cells that line the spongiocele and canals? Choanocytes, also called as collar cells. The spongiocele is lined by flagellated cells called collar cells. Actually, their flagellary beating can set the water currents. Dash can set the water currents, flagellary beating of collar cells. Besides, they also help in digestion of food particles. So the type of digestion is intracellular. That means it takes place within the cell. Type of digestion, intracellular. Okay, ma? Here, respiration by diffusion, excretion by diffusion through body surface. And then here nerve cells are absent, so systems are not developed. Then coming to the sexes, sexes are united, means bisexual. Means monoecious. Very, very, very important. Fertilization is internal. Fertilization is internal. Development is indirect. What is meant by indirect? If any larval stage is present, larva stage is present, it undergo a change called metamorphosis. Metamorphosis. So it passes through different developmental stages. So the type of development is called indirect. If a direct young one is developed, that resembles the adult. So such type of development is called direct. Direct and indirect development. So here in porifera, development is indirect. Reason: larval stages are present. And one more point is given. Here in these animals, asexual reproduction by fragmentation. That means small parts can cut off and each piece can develop into complete organism. This is called fragmentation. Example, cycon, scypha, other name, spongilla, freshwater sponge, use spongia, bath sponge. Why? Because here the skeleton is having a fibers, sponge in fibers. So the sponge is very soft and used for bathing purpose, used as a scrubber. So this is about porifera. Now, Levin. Now
Yes,
tell me, what are the sequence of points generally we'll discuss in each phylum? You tell me.
so it's introduction,
Very good.
habitat, habit, internal features.
Levels of
Oh,
organization,
sorry. Level of organization, internal
symmetry,
symmetry,
coelom,
coelom,
external,
external features,
internal,
internal features.
and physiology. I think all these activities are physiological activities: locomotion,
Yes.
digestion, respiration, circulation, excretion, nervous system, sense organs, and followed by reproduction, development. Finally, examples. Now, you tell me, what is the habitat of sponges?
So they are mostly marine, some are
Very good. And
freshwater.
few are freshwater.
Freshwater.
Now, name the freshwater sponge.
Spongilla.
Very good, spongilla. And then habit.
So they are sessile.
Sessile. Sessile means stationary.
They are
It cannot,
stationary.
move from one place to other place. Okay, ma? And then next, what are the next levels of organization?
So they are cellular.
They are cellular level. Next, asymmetrical.
They
Next,
are asymmetrical,
asymmetrical.
asylum.
Very good. What is the unique feature found in porifera?
So canal system or
Very good. Or it's
transport of
also called
water
as
system.
water transport
Water
system.
transport system.
Now, what are the functions of water transport system? Number one, collection of food. Number two, respiratory gas exchange. Number three, elimination of waste materials. Now, water leave.
So can you just repeat those, sir?
Yes, food collection. Number two, respiratory gas exchange. Number three, elimination of waste materials.
Excuse me, sir.
Yes.
Sir, can you just explain what are these spicules found in
Yeah,
sponges, sir?
spicules are nothing but small spines. Small spines are called spicules, made up of calcium carbonate. So in some sponges, the skeleton, what is the purpose of having skeleton, ma'am? To give support to the animal. Like our skeleton made up of bones. Their skeleton is made up of small spines called spicules. Small spines called spicules, made up of calcium carbonate. In some animals, the skeleton consists of fibers made up of spongin fibers.
So can you explain this again?
Okay. So in body wall, we can find skeleton made up of small spicules. What are spicules? Spines. Small spines are called spicules. They give support to the animal. As this structure gives support to the animal, the name given as skeleton. In some sponges, the skeleton constitute the fibers fibers made up of spongin. So the name given as spongin fibers. Spongin fibers.
Excuse me, sir.
Ma.
Sir, what's the other name of collar cells, sir?
Collar cells, choanocytes. Why the name collar cell is given? See, here there is a collar-like structure. As they are having collar-like structure, the name given as collar cell. Now, what are the main functions of collar cell? Number one, they create water currents by their flagellary beating. Number two, they perform the process of digestion. As the digestion takes place within the cell, the name given as intracellular. What is intra? If a digestion takes place within the cell, it is called intra. If the digestion takes place outside the cell, it is called extracellular. Intracellular and extracellular. So digestion is intracellular. Sexes are united. Fertilization internal. Development is indirect. And in some, asexual reproduction by fragmentation. Example, cycon, euspongia, and spongilla. So this is about porifera. Do you have any doubts? Shall I continue? Right, Anna. Then I'll ask questions. Right. So, Ritvik,
Sir.
can you hear my voice?
Yes, yes, sir.
Now, you tell me. What is the level of organization found in porifera? Cellular level.
I should tell, yeah, sir.
And the skeleton of a sponge constitute dash.
Canal system.
Huh? No, spicules or spongin fibers. And then what is the other name of choanocytes? Collar cells. That means you are not paying concentration. Derwin. 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 Is not responding. Derwin, can you hear my voice? Right, Emma. Chalisha,
Yes, sir.
now tell me, what is the type of fertilization in porifera?
Fertilization, sir.
Fertilization. Internal fertilization. Type of development.
Indirect, sir.
Indirect development. What is the habit?
Habit. All are sessile.
Sessile means stationary. And mostly marine and freshwater. What is the unique system found in porifera?
Water transport or canal system.
Very good. And what are the functions of canal system? Number one, collection of food.
Food, respiration.
Number two, respiratory gas exchange. Number three, elimination of
Waste material.
Waste material. Right, Amma. Again, I'll ask questions tomorrow.
Yes, sir.
And you must come with full preparation.
Okay, sir.
Okay, right. Next, coming to cylindrator. My pen is not working properly. Okay. Right, Emma. Next, cylindrator. Cylindrator. Other name of cylindrator, nidaria. And we'll follow the same sequence that we followed for sponges. Now, Ritvik, you tell me the first point.
So,
what is the first point in sequence?
Introduction.
Introduction. So here, introduction, they are commonly called as nidarians. Why they are called nidarians? I'll tell you. They are commonly called as nidarians. Next point. Habitat. Same, mostly marine. Mostly marine. Few freshwater. Mostly marine, few freshwater. Coming to habit, maybe sessile or free swimming. Maybe sessile or free swimming. Look at the textbook. In last lines, we have two body forms. Polyp, cylindrical shaped structure called polyp. And umbrella shaped structure called medusa. Polyp and medusa.
So polyp is which shape, sir?
Polyp is cylindrical shape and medusa is umbrella shape. Polyp and medusa. Polyp is cylindrical shape. Cylindrical shape. Whereas Medusa is umbrella shape. Now, very, very important, polyp is sessile form, whereas Medusa is free swimming form. And there examples are given. Example for polyp form, Hydra, Adamsia. Adamsia is also called as a sea anemone. Yesterday only we discussed about this. Sea anemone exhibit dash type of symmetry, biradial symmetry. An example for Medusa, Aurelia. Aurelia, Aurelia, commonly called as jellyfish. Commonly called as jellyfish. Jellyfish. First point, they're commonly called as cnidarians. Number two, mostly marine, few freshwater, maybe sessile or free swimming. Next, after this, levels of organization, tissue grade of organization. Number two, diploblastic. Diploblastic. What is meant by diploblastic? Two germ layers. What is the type of symmetry? Radial symmetry. Radial symmetry. Radial symmetry. And A, coelomates. A, coelomates. I think some problem is there with pen. Anyway, I'll explain the points with spellings. Just don't worry. Number one, most commonly called as cnidarians. Number two, they are mostly marine, few freshwater. Number three, they may be sessile or free swimming. Example for sessile, Hydra and Adamsia. What is the common name of Adamsia? Sea anemone. Maybe free swimming, umbrella shape. Example, Aurelia, jellyfish. Next point, levels of organization, tissue level, diploblastic, radius symmetrical, acoelomates.
Excuse me, sir.
Ma,
Sir, what's the other name of Adamsia?
sea anemone, sea anemone. Next, coming to the body structure. See, these are the true diploblastic animals having two germ layers. What are the two germ layers? Ectoderm and endoderm. Between dash is present
Mesoglia.
Mesoglia. Very good. Mesoglia is present. Mesoglia. Look at here. Diploblastic consists of two germ layers. And here the body wall encloses a cavity called gastrovascular cavity, or simply called as GVC. Gastro means help in digestion, vascular, help in distribution of food, cavity. Or cylindron. Gastrovascular cavity or cylindron. And this gastrovascular cavity also lined by flagellated cells like sponges. They also perform the process of digestion. And this gastrovascular cavity opens out through mouth. Very, very, very important. The mouth is located on a small conical structure, what we call it as hypostome. 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 HYPO. Hypo means low, stoma means mouth. Hypostome. Hypostome. And surrounding the mouth, we can find tentacles. These are all tentacles.
Sir, can you explain this hypostome again?
Okay, I'll repeat once again. Right. So this is body wall, encloses a cavity called gastrovascular cavity, lined by flagellated cells. And this gastrovascular cavity opens out through mouth, and the mouth is located on a small conical structure called hypostome. Hypostome. The mouth is located on hypostome. That sentence was given in NCERT. Mouth is located on hypostome. Hypostome. Okay, ma? See, hypo means this is mouth. The point or the area below mouth is called hypo. Hypo means below. Stoma means mouth. The area which is found below the mouth is called hypostome. That is surrounded by tentacles. Tentacles. And very important. Here the tentacles and body wall consists of special type of cells called nidoblast cells. They are called Nido, CNIDOBLAST, nidoblast cells, with a sac called nematocyst. What is that, Amma? Nematocyst. Nematocyst. Nematocyst.
So where are these nidoblasts present?
Nematocyst. Amma?
So where are these nidoblasts present?
All over the tentacles and also on body wall. All over the tentacles and on the body wall, we can find special type of cells called nidoblast cells, having a sister or a sac-like structure called nematocyst. And here we can find different types of nidoblast cells. In some, we can find poisonous material. With that poisonous material, they can kill the prey. Actually, they catches the prey with the help of tentacles and they inject the poison through thread-like structure. They just penetrate these threads or needle-like threads into the body of prey. They inject the poison, they paralyze the prey, and they feed. Some nidoblast cells are having a sticky substance, gummy-like substance, which help in attaching to the substratum. What is meant by substratum? Any object. So, what are the functions of nidoblast cells? Look at the textbook. Number 1, food capture. Number 2, defense. Number 3, anchoring. What is meant by anchoring? Attachment. Anchoring means attaching to the substratum or any obstacle. Anchoring means attaching to substratum. So functions of nidoblast. Number 1, food collection or catching the prey. Number 2, attaching to the substratum. Number 3, defensive mechanism. That means as they are having a poisonous cells, they can protect themselves from other animals.
Sir, can you repeat the functions?
Yes. Number 1, food collection or catching the prey. Number 2, anchoring. What is meant by anchoring? Attaching to the any obstacle or any object. Number 3, defense. Help in both offense and defense. So external, internal completed. Coming to digestion. Here digestion is both extra and intracellular. First, digestion takes place in GVC. Amma, what is GVC? Gastrovascular cavity. And partly digested food is taken up by the flagellated cells and they finish the digestion. The first digestion takes place outside the cells in cavity, called as extracellular. Later on, the digestion will be completed within the cell. It is called intracellular. So the digestion is both extra and intracellular.
Sir, can you repeat?
Okay. digestion is both extra, takes place in GVC. How the digestion takes place, I'll tell you. First, it catches the prey and just takes into the body through mouth. Once the food reaches the cavity, we can observe the release of some enzymes. And those enzymes can finish the partial digestion. And partly digested material can be taken up by the cells and they finish the remaining digestion. So final digestion takes place within the flagellated cells. Whereas in case of sponges, remember the digestion is only intracellular, carried out by choanocytes. They are carried out by choanocytes. But here, first digestion takes place in GVC and final digestion takes place within the cell. So the type of digestion is both extra and intracellular.
Sir, I have a question, sir.
Yes.
Sir, this GVC, I mean, this cavity is present inside the body, right? So how is
Yes,
it
inside the-
extracellular digestion?
Yes, I'll tell you. Look at this amoeba. Amoeba just takes a food material into the cell as a food vacuole. Into this, digestive enzymes are released by the fusion of lysosome and digestion can be completed within the cell. This is called intra. Got it? Now look at this. This is hydra having two layers, ectoderm, endoderm. Endoderm is having dash type of cells, flagellated cells. Now the layer encloses a cavity. Just remember, just try to recollect our elementary canal. This is our elementary canal, isn't it? And the wall is formed by cells, isn't it? Within, you can find a space called lumen. Now, generally, we'll take the food and that food passes through esophagus, stomach, and intestine. While the food is passing through the cavity, I'll take a cross section of this gut. See, this is the gut or elementary canal. It is made up of what? Cells. Okay, ma? Okay, now this is called lumen. What is this lumen? Cavity. Now, once the food reaches the lumen, you can observe the release of enzymes into the space. Now, by the action of enzymes, the food can be digested. So, where does this food digestion takes place, you tell me? Outside the cells, within a cavity. I think you got it. Digestion takes place. Amma, look at here. All these are cells, isn't it? All these are cells. Where does this digestion takes place outside the cells? In a cavity. In a cavity. So the type of digestion is called extra. Outside, but between the cells. Outside, but between the cells. And partly digested food is taken up by the flagellated cells and the final digestion takes place within the cell. This is called intracellular. Okay, ma? I think you got it now.
Yes, sir.
Very good. And then next, you can add one more point. Already we discussed that. Nerve cells first appeared. Nerve cells first appeared. Nerve cells first appeared. And there a point was mentioned. Already we discussed this. Some animals are cylindrical. Some animals are cylindrical. Some animals are cylindrical. And some are umbrella shape. Example for cylindrical. Cylindrical forms are usually sessile. Amma, what is meant by sessile? Stationary or fixed. And these are free swimming. Example, Aurelia, commonly called as jellyfish. Example for sessile forms, Hydra and Adamsia. And very, 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 very important. In some animals like Obelia, very, very important. Two forms are found in the life cycle. In one generation, we can find sessile polyform. What is this form? Polyform. This is free swimming medusa form. Very, very important. In Obelia, the polyform give rise to medusa by a process called budding. It's an asexual reproduction. And the medusa produce gametes. Those gametes fuse and form larva. And again, they give rise to polyp. So in one generation, we can find asexual reproduction. In another generation, we can find sexual reproduction. This is sexual. This type of development is called, very, very important, alternation of generation or metagenesis. Alternation of generation or metagenesis. Very, very important. This type of alternation of generation is found in Obelia. And this alternation of generation or metagenesis is not found in all cnidarians, found in few cnidarians like Obelia. Okay, Amma? I'll repeat once again. So here we can find two types of body forms. Cylindrical sessile polyform, umbrella-shaped free-swimming medusa form. See, some cnidarians are polyform. Some cnidarians are in medusa form. But some show both forms in their life cycle. Best example, Obelia. In one generation, we can find asexual form that give rise to medusa by asexual reproduction. And medusa produce gametes, they fuse and form larvae that undergo development to become polyp form. So in one generation asexual form, in another generation sexual form, this is called alternation of generation. For every generation, you can observe alternation of forms. This is called alternation of generation or metagenesis. Okay, ma'am? And one more extra point, very important. This is there in MCRT. Some animals produce calcium carbonate exoskeleton. What is exoskeleton? Present around the body. The calcium carbonate exoskeleton, they are called corals. They are called corals. Coral. I think you might heard the term coral reefs. Coral reefs. Best example, Lakshadweeps. So from millions of years, what happened? They go on secreting the calcium carbonate, and this was deposited and come to the surface of the sea and developed as islands. I think you might have seen red stones. Actually, in middle of the ring, you might have seen green red stones, isn't it? And these green red stones are nothing but corals. Next up.
Sir, can you
Sexes.
explain this again, sir?
Yes. Some animals, some cnidarians, they continuously secrete or produce calcium carbonate exoskeleton for protection. Those secretions, they go on and finally becomes islands called coral islands, coral. The calcium carbonate exoskeleton of anthozoans or cnidarians is called coral. I told you, black, red color stones which are found in rings are nothing but corals, coral stones.
Sir, so this exoskeleton is called coral, sir?
Yeah.
Okay, sir.
Sexes united. Fertilization external. Development indirect. Development indirect. Look at the textbook. Example, Hydra, Obelia, Physalia, commonly called as Portuguese man of war. Adamsia, sea anemone, Pinnatula, Pinnatula, sea pen. That means it looks like a feather. In olden days, we used to write the things with the help of feather. So it looks like feather, so the name given as sea pen. Gorgonia. Look at the textbook for these spellings because my pen is not working properly. Gorgonia. Sea fan. It looks like a paper fan. So the name given as sea fan. Meandrina. Brain coral. Meandrina brain coral.
Excuse me, sir.
Ma.
Sir, this nidoblasts are the stinging cells, sir.
Yeah. They're also called as stinging cells. Why? Because they just pierce through the skin of prey and they inject the poison. So the name given as stinging. Okay, ma?
Sir, so then what are these nematocysts, sir?
Nidoblast is the name of the cell with a thread-like structure called nema. Nema means thread. So the name nidoblast is having a cyst or a sac called nematocyst, which is filled with either poison or sticky substance. As they can pierce through the skin of the prey and they can inject the poison to paralyze the prey. So the name given as stinging cells. So nidoblast cells are commonly called as stinging cells.
Sir, so they are the cells which have these nematocysts.
Yeah. Original name nidoblast. They help in stinging, so the name given as stinging cells. Having a cyst called nematocyst. Nematocyst. So this is about Nidaria or Cylindrata. Now you tell me, what is the level of organization? Tissue level, diploblastic, radially symmetrical, acidomates. The body wall encloses a cavity called gastrovascular cavity or cylindron open out through mouth located on hypostome. Mouth is surrounded by dash tentacles. Tentacles and body wall consists of dash nidoblast with a cyst called nematocyst, which help in food collection, defense, and anchoring. Type of digestion, both extra and intracellular. Okay, ma? Nerve cells first appeared. What are the two main forms found in these animals? Either polyp or medusa. Dash is the fixed form, polyp form. Free swimming form, medusa form. So some animals may be either polyp forms or medusa forms, but some show both the forms. Best example, obelia, where one form alters with other form in different generations. This is called alternation of generation. Other name of alternation of generation, metagenesis. Example, obelia. Okay, ma? Some produce calcium carbonate secretions. They form protective exoskeleton. Those exoskeletal structures are called corals. The largest coral is Great Barrier Reef of Australia. Great Barrier, just you search that Great Barrier Reef of Australia in net, you can see everything. You can see all these corals, what are coral islands, how they are formed, everything you can get in Google search. Right now, next phylum, Ctenophora. This is the last phylum of the day. Ctenophora. Ctenophora. Same. Look at the textbook and you can see each and every line. First, what is the first point while we're studying the characters? Number one, introduction. They're commonly called as sea walnuts. Sea walnuts or home jellies. Home jellies. Sea walnuts or home jellies. Sea walnuts or home jellies. Habitat. Very, very important. All are marine. All are marine. All are marine. All are marine. All are free living. All are free living. Coming to levels of organization, same. Tissue level of organization. Diploblastic. Radially symmetrical. Acoelomates. Tissue level, diploblastic, radially symmetrical, acoelomates. And one very important point, they are close relatives of Cnidaria. Close relatives of Cnidaria, but Cnidoblast absent. So the name given as Cnidaria, Cnidarians or Cnidarians due to the absence of Cnidoblast. Due to the absence of Cnidoblast. Next, coming to the body structure. You can see the diagram in textbook because my pen is not working properly. All over the body, 8 rows of ciliated comb plates are present. I'll show you one comb plate. See, This is one comb plate. Many cilia are arranged in such a fashion that it appears like comb. Okay, Amma? What is comb? Row of cilia. Cilia. A row of cilia. And these are called comb plates. These are called comb plates. How many rows of comb plates are present? 8 rows. They help in swimming. And of course, they also having a pair of tentacles. Useful for food capture, not for swimming. Useful for food capture. Useful for food capture. And very, very, very important, they show a phenomenon called bioluminescence. What is meant by bioluminescence? Emission of light. You can watch these pictures in net. Bioluminescence, they emit light. Bioluminescence is the characteristic feature of ctenophores. I'll repeat once again. Commonly called as sea walnuts or comb jellies. Complete marine, complete free swimming, tissue grade, diploblastic, radially symmetrical acoelomates. Like Cnidaria, they are diploblastic. Like Cnidaria, they exhibit radial symmetry. Like Cnidaria, they have extra and intracellular digestion. Very important. Digestion is both extra and intracellular. So they show similarity with Cnidarians in many aspects except Nidoblast. So the name given as Cnidaria or Cnidarians. Type of digestion, both extra and intra. Sexes united, means bisexual. I'm a very, very, very important here. No asexual reproduction, only sexual reproduction. Only sexual reproduction. Fertilization external and development is indirect with larval stage. Example, pleurobrachia and pleurobrachia and tenoplana. Pleurobrachia and tenoplana. Pleurobrachia and tenoplana. And what are the unique features found in Ctenophora? Number 1, bioluminescence. Number 2, arrangement of comb plates in how many rows? 8 rows, who help in swimming. This is about Ctenophora. And tomorrow, I'll ask questions from all these phyla. Okay? Are you following my teaching? Amma?
Yes, sir.
Okay. You do one thing, you just tomorrow, all of you try to come with full preparation. And I should see each and every one tomorrow. I'll ask each and every one. But some students, they are not turning on their videos. I don't know what is the problem. Gervin, I did not see you right from first class. And number two, right, okay. I think Prithvi, you said that his network is not proper.
Yeah, so it was before.
Okay, okay, right. Right, Emma? Is this pattern of teaching is okay for you?
Yes, sir.
Are you following my teaching? Right, okay. So tomorrow, I'll ask questions individually. Okay, Emma? Get
Yes.
ready for that and have a nice day. Good night. Thank you. Thank you very much.